Good evening, everybody. Good to see so many people here at the fundraising dinner. My name is Ernst Wolvetang. I'm a professor at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. And I'm talking to you tonight about a plan how to develop therapeutics for CASK-related disorders, which is why you're all here tonight. So let me explain a little bit what my lab does and what we are thinking, what we can do for this group of disorders. As you probably all aware, CASK-related disorders are due to mutations in the CASK gene, which is a very important protein that has important functions in the brain and for brain development as well. And at the moment, really, we don't exactly understand why these disease phenotypes, so these disease problems occur in our children and what we can do about it. So how do you start that? Well, one of the plans that we have is to make brain models from blood cells. And let me explain to you how that works. So what we are able to do is to take a small amount of blood from children and their parents and reprogram those cells, those blood cells, back to stem cells that were the same when you were a day 5-old embryo before you were implanted into the womb of your mom. And that does two important things. Firstly, it captures the genetic makeup of the individual. So in this case, the genetic makeup of the children that we are trying to treat in the future. And it allows these stem cells to make all the cell types of the human body. And in this case, we're particularly interested in the central nervous system, obviously. So what we do in my lab is once we have made these stem cells from the blood cells from the individuals that we are trying to treat, we can convince these cells to become neurons and glia, the main cells that make up our brain. But we can also make what's called brain organoids, which are three-dimensional representations of different regions of the brain, and that over time develop also the function of the brain. So we have been using this model for a whole number of neurodevelopmental and neurodegenerative diseases already, like for example ataxia telangiectasia and hereditary spastic paraplegia. So we have a lot of experience in using these models to not only find out what is going wrong in a particular disease that is caused by mutations in the DNA, but we also have used these models to identify where and when things go wrong in the brain, and then to use those models to start screening for therapeutics. And these can be either drugs or gene therapy, where we try to replace the defective gene with a functional gene. Now, for CASK and CASK-related disorders, we are still very much at the very start of this journey. But the fact that we've been able to take these models from other neurodevelopmental diseases into now clinical trials for children with ataxia telangiectasia, for example, suggests that we can actually do this and actually can make an impact for people and their families. So let me explain how we would do that. We generate these stem cells, we make brain organoids, and then we have automated robotic systems that can screen a whole range of drugs and to see which one are effective. And we can also use those to test whether replacing the gene is a good way to go. So as I said, we are still at the start of the journey, but this research is of course expensive, and we need to employ the people to do this. So what I'm hoping is that you can look into your soul and into your wallets tonight and contribute what you can, so we can start to make the first models of the children that have CASK and CASK-related disorders. This is, as I said, this is already a pipeline that we know can work and could actually make a real impact for children down the track. So I am hoping that you can contribute to this journey and to this mission. And your contributions are really, really important, because the research programs that we run are in many cases supported by government funding from the Medical Research Future Fund. And what that fund requires is both a co-investment by the patient advocacy groups, as well as co-design of the research together with the people that we're trying to treat and their families. And this is the first step on that journey. So what I'm hoping that we can do together is collect money, establish the models, get the data to show that this is the right way to go, and then write grants for the federal government and see if we can get a program off the ground so that we can do this at scale for both our Australian patients, but also for all the international patients with CASK. So with that, I'm going to let you get back to your dinner and your after dinner drinks, but I hope I've given you an insight into how small steps can lead to really big outcomes down the track. And that stem cell based modeling with patient-specific models of the brain are a really powerful way to move forward towards therapeutics for this devastating disorder. So with that, I will let you go and wish you goodbye. Bye bye.